So today I'll use the psychological model of addictions that I've developed over the last 3 years with Barry Everett that we've called incentive habits, to emphasize that addictions perhaps are much more complex than we initially thought, that they might have nothing to do with the ventral striatum, perhaps nothing to do with dopamine. No, I'm kidding. There's something to do with that. But the point is it goes beyond that. And before I dig into addiction, I think the take-home message here is I would like you to really appreciate that behavior is a technological tool as much as optogenetics or whatever transgenic model you can have. Even though usually you consider behavior is just something to screen the molecular and biological tools you're going to use, I think in the field of neuropsychiatry, it is the core and the most important technological tool you have to use. And I've slipped into my talk 3 slides to actually follow what Nora Volkov discussed about yesterday about compulsivity and OCD, and she put in parallel OCD and drug addiction. I just wanted to show you that you can also study OCD and the compulsive nature of OCD in parallel with the compulsive nature of addiction in rats, in preclinical models. So if you look at the definition of OCD in the DSM-4/5, it's characterized by repetitive behaviors that are aimed at reducing distress or preventing some dread situation, that are excessive or unreasonable, interfering with activities, and they actually are responsive to SSRI, serotonin reuptake inhibitors, antidepressants. Even though actually the dose you would give to someone who's suffering from OCD is in average twice the one you would give to someone suffering from depression. And in animals, the schedule-induced polydipsia model, I'm going to tell you what it is later on, is a repetitive adjunctive-inducing behavior that is anxiolytic. It is excessive, unrelated to thirst, rigid and maladaptive, and sensitive to SSRI. Basically what polydipsia is, for those of you who like beef, I serve you a nice beef steak like that, but I give you 1 milligram per minute. Are you going to like it? I guess you won't. Actually, you want a full mouth of it. You want to have it the way you like to have it. So that's exactly what we do. The animals are placed in a box. They know food is coming, but they have no control whatsoever over it. It's coming, but they can't have it the way they want. This generates frustration to which the animal is going to cope with expressing a compulsive maladaptive behavior on something on which they can exert control in the environment, that is a bottle of water. There's water there available, and while one rat would drink 5 milliliters per hour to actually to cope with thirst, in face of this adverse consequence, adverse situation, the animal can drink up to 50 milliliters per hour. So a rat of 350 grams would drink 50 milliliters per hour. I'm 100 kilos, so we're doing 35 liters per hour. So we've got to stop them because they're killing themselves. Okay? What's interesting is I've emphasized that because I'm going to discuss about it today. We've been interested over the last years in the relationships between impulsivity and various behavioral expressions of compulsivity, as you can find in different obsessive compulsive spectrum disorders. And we can identify high impulsive animals. I'll come back later on the way we identify them. But nevertheless, high impulsive animals. And in the schedule-induced polydipsia procedure, in other procedure, we can identify high compulsive rats. And we've been investigating whether high compulsivity could be predicted by high impulsivity. And the point is, yes, high impulsive animals here, when you put them in the schedule-induced polydipsia procedure, they develop drinking levels that are much higher than low impulsive animals here.
Just a quick question.
Yep.
What is the difference between impulsivity and compulsivity?
So in the DSM-4, compulsivity is a repetitive behavior that aims at reducing distress. It is an anxiolytic behavior. You do something, you don't really like doing that, but once you've done it, you feel relieved. Impulsivity is the opposite. There's something in the environment, you have an aberrant reaction to it, and you feel guilty once you've done it. Yeah? So impulsivity, there's no foresight of what you're just going to do, and you have no idea about the consequences, or you shun them off if you have any idea. Compulsivity, you plan. You know what you're going to do is not actually adaptive, but you know you're going to feel better once you've done it. Yep? So that's the definition in the field of OCD. In the field of addiction, compulsivity is maintain drug use despite knowledge of adverse consequences. Yep?
So how would you, and we can talk about this later if you want, but how do you fit in risk-taking behavior in this? Because we also see in addicts that they engage in like high elevated risk-taking behavior.
Yeah. So if you don't mind, we'll come back to this question at the second part of the talk. But definitely that's very important. We'll come back to that. Well, I want, yep.
Sorry, just one more.
Oh, go ahead.
So I guess those definitions seem kind of opposing, impulsivity and compulsivity. In the sense, impulsivity seems like a rapid switch, or at least they seem like rapidly switching between environmental stimuli or whatever it might be.
It's not rapidly switching, it's inability to refrain from maladaptive prepotent responses. Yep. Because it's absolutely, you can double dissociate impulsivity from flexibility. So the point is, so low impulsive animals would develop polydipsia, but not that much. High impulsive animals would develop high levels of polydipsia. Now, if you give chronically atomoxetine, a noradrenergic selective reuptake inhibitor that has been used successfully to treat ADHD, so it decreases impulsivity, but so far it's never ever been used in OCD. The point is, atomoxetine decreases impulsivity only in high impulsive animals. It has no effect in non-impulsive animals. Okay? And it also decreases the emergence of compulsive behavior and the overall level of drinking in high impulsive animals. So it has an effect on both impulsivity and the propensity to develop compulsive behavior subsequently. Interestingly, if it had an effect on non-impulsive animals, it would be the opposite. It would increase compulsive behavior, even though this effect doesn't reach statistical significance. Now, what I would like to point out is if we had considered the effect of atomoxetine on a cohort of animals that would develop compulsivity, add this effect to this effect, the result is no effect of atomoxetine. And the point is, from a clinical point of view, you don't want to treat these people. You're going to see them. These people aren't suffering from any psychiatric disorder. These rats, but these aren't compulsive. So those you want to check whether they would respond to a specific treatment are these ones. And digging into inter-individual differences helps you bringing effects of different drugs that you wouldn't find otherwise if you were focusing on cohorts of animals. Okay?
Can I just ask, so the high versus low impulsive rats, are those two separate colonies that you're breeding, or you're screening them?
So we always, so now in Cambridge, so these animals have been developed in Cambridge. Yeah. Jeff Belin, 2007 in Science, and Jeff has now bred them. I'm not using that, so he does it. I'm not, because I think when you breed and when you enrich a specific trait, you bring with the breeding only the polymorphism and the genetic background. And if you use cohorts, you bring everything, the story of the animal in the cohort, everything. So I always screen animals from a cohort of 48 rats, always, and upper lower quartile. I don't do median splits, because in that case you would introduce type 2 errors, so upper lower quartile. And when you reach a number of animals that is big enough such that you can say this one is an high impulsive because his behavior is above X level of responding, then you don't have to split with upper lower. You just say this is impulsivity, this is not. And I think now we reach the stage where we have 3,000 rats, so we start knowing that above 50 of premature responses, the animal is 100% impulsive, and below 20, the animal is 100% clinical percent. Yep. So it was just to actually to discuss what Nora presented yesterday. Yes, compulsivity is something that is not specific to addiction. It spreads through many neuropsychiatric disorders, even though we call compulsivity something in addiction that has psychologically nothing to do with the compulsivity we would find in obsessive compulsive disorder. Because in obsessive compulsive disorder, compulsive behaviors are excessive maladaptive behaviors that aim at reducing distress. George Koob and Michel Lemoyle has put a parallel between this compulsivity and their definition of compulsivity in addiction, that is, negative reinforcement driven behavior. But the point is, once you've gotten rid of the aversive properties of withdrawal, it would still relapse, and you would still compulsively relapse. So the point is, compulsivity in addiction, perhaps there's definitely a component of withdrawal, but the compulsivity is not withdrawal related necessarily. Okay? So let's start discussing about addiction now. So addiction is not about drug taking, because if you look at the criterion, the DSM-4, so initially, addiction was defined as tolerance and withdrawal. This definition has lasted until 1987. That was addiction, because at that time, most of the drug addicts were opiate addicts. And definitely, opiate addiction has to do with tolerance and withdrawal. I'm not going to discuss about that at all today, because I don't really care about that. These are biological adaptations to drug exposure. This is pharmacology. And you can have tolerance and withdrawal to many drugs to which you would never develop addiction. Okay? Then in 1987, were introduced several criteria that had nothing to do with pharmacology, more to do with compulsivity. And in 1994, and the DSM-4, appeared the criteria that are now common to us, which are substance used in larger amounts or over a longer period than the period intended, persistent desire or one or more unsuccessful effort to cut down or control substance abuse, important social, occupational, or recreational activities given up or reduced because of substance use, a great deal of time spent in activities necessary to obtain, to use, or to recover from the effects of substance use, and most importantly, continued substance use despite the awareness of the adverse consequences of use. Okay? This is far behind withdrawal or tolerance or whatever. And if you want to psychologically put everything together, you could say that what matters in addiction is craving. Craving is an obsession, the same kind of obsession you would have in OCD. It's an idea that comes into your brain, that spreads, and that kills everything else. And if you want to fight it, you've got to recruit all your cognitive resources and fight it. You would eventually relapse, or you would win against it, but it's there. And this obsession is highly motivational and is related to drug seeking. But the point is, craving doesn't necessarily precede relapse. And there's other components in addiction that are important, and I'm going to focus on the notion of compulsive habits. Okay? And now if we try to operationalize the different stages of drug addiction, you've got initiation of drug use, recreational drug use, 99% of the field works on that. Then habitual compulsive drug use, abstinence, relapse, and you end up with cycles in between them. And this is addiction. Addiction is there. And over the last 7 years now, I've been trying to operationalize the DSM criteria in preclinical models that we're going to discuss about today. And most specifically, the high motivation for the drug, loss of control over drug intake, maintained drug use or relapse despite adverse consequences, and I'll give examples of that, Q-controlled drug seeking. And we'll discuss about drug taking, drug seeking, what is the difference, why would we want to measure drug seeking and not drug taking, and the other way around. That's very important for us. what's important for us is this guy has just been arrested and is asked to blow into this machine. And that's what he does. I'll put that once again. He's just been arrested. Please, can you blow in that? And the guy said, yes, of course. Up. Okay, so let's say if it had been alcohol in the bottle, the guy would have relapsed. Okay? I bet you nobody would tell me here that the guy craved before. This is a typical example of a reflex. I have a bottle, I drink. There's no craving, but there's relapse. Okay? And now put this behavior in front of some results we've had from clinical studies. So Nora demonstrated in 2006 that cocaine addicts, they're in the PET scan like that. They just don't move. And they're presented with images that are related with cocaine taking behaviors. And they crave. And there's a massive increase of dopamine transmission into the dorsal parts of the striatum. This part of the striatum that has to do with Parkinson's disease, Huntington disease, but not much with motivation. So how can you link this desire for the drug, so powerful that is craving, with dopamine transmission into the dorsal areas of the striatum? One potential interpretation is neurobiological, another one is purely psychological. This is not a correlate of craving. It could not be. It could be something else than a correlate of craving. If I'm a drug addict, and I'm in a PET scan like that, and I'm presented with stimuli that drive my automatic behavior towards taking the drug, but the drug is not there. Nevertheless, all the neurobiology driving my habit has been implemented, and then there's a mismatch between what has been implemented and the environment. And this mismatch, then retrospectively, I start craving because there's no drug there. So it would be very interesting, I'm not sure we could do that, to provide these addicts in the PET machine with cocaine. They've got cocaine here, we present them the image, they take cocaine, and I'm pretty sure you will have no dopamine release in the dorsal striatum, and no craving. Or increased dopamine in the dorsal striatum, no craving, but they would still take the drug.
So you could also say it's a reward prediction.
It goes beyond that, yeah. So that's the point. Addiction, perhaps. I'm talking about actions in terms of goal-directed instrumental responses. I'm going to discuss about habits in terms of rigid, non-goal-directed behaviors. So in terms of psychological processes, I promise then there's going to be neurobiology. But what's really important is so far, all the research worldwide, all the therapeutic strategies, all the CBTs, cognitive behavioral therapies, are built upon this model of addiction. All of them, which is through repetitive conditioning and repetitive associations between the effects of my drugs and the environment. I've learned that some exteroceptive stimuli drive my incentive learning processes to an aberrant motivation towards the drug, okay? And this aberrant motivation can't be stopped by a failing inhibitory system, that is my disrupted prefrontal cortex, such that I crave and I seek my drug, and everything happens within the explicit level of the brain. I know everything. No, there's the stimulus, I want my drug, my inhibitory system can't prevent me from going and seek my drug, and I'm going to take it. Now, what if, instead of happening that way, sometimes actually everything starts the same way initially, but then the motivation is beyond the explicit level. You're not aware of your motivation. It's an implicit urge. This implicit urge, you're not aware of it, drives an automatic behavior which you're not aware of. You would relapse, not even being aware of what just happened. Once you've relapsed, obviously, you would have a retrospect analysis and say, yeah, I wanted it. Otherwise, why would I be drinking my alcohol? If this is the case, then all the strategies aiming at restoring this miss the point. There's a big difference between being able to stop what you see and not seeing what's happening. How can you control something you're not aware of? So this raises a question. Can you have non-explicit motivation? Do you have any examples? Can we show that we can be controlled by urges we are not aware of? Damaso and Béchard demonstrated that quite well, but there is a very nice seminal study by Ken Berridge, who took these people to the cinema, 3 cohorts of people. They're going to watch the same movie, except the first cohort within the movie are presented with subliminal images that are neutral. Nature, whatever you want. So let's say the 3 cohorts are males, and the second cohort is presented with pictures of underwear or whatever you want, and the third cohort is presented with pictures of car accidents. They watch exactly the same movie. They are not aware of the subliminal images that have been presented. They leave the room, and they're asked, okay, so how was the movie? The first guy said, yeah, fair enough. The second guy said, oh, great, really good. And the third guy said, no, I didn't feel good about that. And then there's a drink. And those people who've been presented with subliminal images that were neutral, they would have drink, interact with people, that's the baseline. Those who were presented with positive, enhancing images would seek more social interactions, would drink more orange juice, and those that were presented with aversive images would actually present a complete inhibition of interactions. Some emotions that were driven outside the explicit field could completely control the motivation for social interactions or drink or whatever you want subsequently. And even the subjective report of the affective evaluation of the movie. So we have emotions outside the explicit field. And these emotions or impulses, if they are maladaptively linked to automatic processes, could bridge impulse to behavior without any explicit awareness. And I'll try to demonstrate to you that we now reach the edge of the understanding of how it can work in rats. So first of all, addiction, I told about that, discussed about that yesterday, has to do with conditioning. There are different kinds of conditioning. The first one that is the most studied is Pavlovian conditioning. Every single time you're exposed to a stimulus that is an unconditioned stimulus, a reinforcer, and happens in the environment stimuli alongside the unconditioned stimulus, you would build up associations between the unconditioned stimulus and those stimuli that become conditioned stimuli. And these conditioned stimuli, CSs, are actually get all the motivational properties of the unconditioned stimulus. They become rewarding by themselves. And interestingly, if you have one rat drinking sucrose and you present a light as a conditioned stimulus, when the rat approaches the light, it'll try to lick the light. Now if you present a light alongside a pellet and the rat eats the pellet, when he approaches the light, it tries to eat the light. So not only does this conditioned stimuli get the motivational value of the unconditioned reinforcer, but they are also encoding the sensory specific properties of the reinforcer. Okay? And when drug addicts are presented with these stimuli, often they crave, and craving has been associated with activation of all the limbic areas of the brain, including orbitofrontal cortex, amygdala, insular cortex, and the ventral striatum. Okay? But actually, if addiction was only Pavlovian, people wouldn't have to seek for their drugs. the drug would come from the sky. Right? You've got to work to get your drug. There's instrumental actions in there to get it and to use it. Okay, so drug use is an instrumental action. Drug seeking and drug taking are instrumental per se. And drug use is relatively fast and efficient. It is readily enabled by particular stimulus configurations. It is initiated and completed without intention. Difficult to impede in the presence of triggering stimuli, and effortless and acted in the absence of awareness. And these features that actually characterize every single drug addict resonate well with the definition of habits that are speed, autonomy, lack of control, effortlessness, absence of conscious awareness. I'm not saying that drug addiction is a habit. No. Habits contribute to the development of drug addiction and support the persistence of the disorder. And when I discuss about habits, I'd like you to remind that I'm not talking at all about skills. Lacing your shoes is a skill. This is a habit. Habits are the psychological processes that support the initiation of a single action, not its performance. Once you reach the stage of performance, you discuss about skills. Obviously, habits and skills develop together, but they can be dissociable. I can repeat that. So often, when people discuss about habits, they consider all the automatic behavior that is part of everyday life. Cycling is a habit, driving is a habit, lacing your shoes is a habit. How that could be, involved in the pathophysiology of drug addiction? Because these aren't habits. These are skills. And skills are partly encoded in the spinal cord. In the dorsal striatum, yes, but there's LTP and motor neurons in the spinal cord that support skills. Not that much habit. Yep.
So where do you put the concept of effort in that? Beginning.
So Dickinson in 1985, when he developed the notion of habit, showed that the way every single instrumental response and its relationship with the outcome is processed in the brain after overtraining or under certain circumstances or schedules is nonlinear. At one point, it's the environment that drives the behavior, so there's no relationship, no representation of the relationship between what you do and what you get. Once you break this relationship, the notion of effort is lost. If I know that, I'm gonna earn 10,000 euros if I press on a lever when I'm in that room, I'm not gonna focus on pressing on the lever. That's boring. I'm gonna focus on something else, and I'm pressing because I know it's gonna come. And there's no notion of effort. It's coming. The environment tells me it's gonna come. So in everyday life, how would you dissociate habits from skills? So it must have happened to some of you. We travel quite a lot. You end up in this hotel, let's say. It didn't happen to me last time, but I end up here. It's 10 to 11 in the night. I enter my room. I open my door. I switch on the light. I get my shower, and I go out. 2 hours later, I'm coming back. I didn't drink much, okay? I don't drink too much. Oh, wait, I forgot something. When I enter my room, I switch on the light. The bulb breaks. I take my shower, I leave. 2 hours later, I come back. What do? And then I say, oh yeah, of course, the light's broken. I didn't forget. Actually, when it's dark and when I open the door, usually I open the door that way, and I do that. And if the switch is here, then, I keep on searching the switch here. No? That's a habit. Typically, the environmental configuration drives a behavioral pattern that is absolutely automatic. And then, once I realize that things go wrong, I can change my behavior, I can regain control over my habit, and I can update the motor sequence and the relationships between the environmental configuration and this motor sequence. This is how life works. And most of our behaviors are habit-driven.
But what is a skill then?
So the skill is when you cycle and when you perform the action, this is a skill. It's how well you perform a specific action. Yes?
I think I'm thinking of what you're saying, and maybe I have this wrong, so maybe you can correct me, but I'm thinking you're saying driving is a skill, and I know I want to go that way,
Yep.
so I'm going to turn right, but I have the habit of always turning on my turn signal. Whenever I turn the wheel, or whenever I have this intention, I don't really think about it, it's just now it's triggered.
So let me answer your question right now. What matters is you can have an overall goal-directed cognitive scheme. I want to go to the lab, driving. Okay, my behavior is goal-directed. Within this overall goal-directed cognitive scheme, I would put together different subroutines that are stimulus-response, change my gears, whatever I want, but when the light turns red, I stop. Yep. Now, I can have an overall cognitive scheme that is a habit. I switch on the light. You know? And within this cognitive scheme that is a habit, I would implement goal-directed actions. Actually, I'm going to change the bulb. I'm not stuck into my habit. I'm going to do things. Or the same thing with, drug addicts. My dealer is not here. I stay 2 weeks, 3 weeks. No, I'm going to actually put together subroutines that are goal-directed. Within an overall goal-directed cognitive scheme, you can implement subroutines, and they can be habits or skills. And actually, when you're supposed to be turning right and you're turning left, this is, at that time, what drives the behavior is a habit, even though you were performing a skill. Because skill is about performance, habits are about initiation of action. Turning is an action. But within an overall habitual cognitive scheme, you would still implement goal-directed actions. The best examples are people suffering from paranoia. The best habitual cognitive schemes ever. My wife was supposed to call me 10 minutes before my talk. She hasn't called. She's with someone else. That's my reaction to the fact that she didn't call me. And this is driving me mad. And when I'm going back home, I try to find the guy and I try to kill him and I beat my wife. Because she's cheating on me right now. But that's people suffering from that's what happens. And you can treat them, making them understand that this scheme is maladaptive and they can regain control over it and change the cognitive response that is rigid to the stimulus that is she hasn't called me. Okay? So habits are not about performance, they're about initiation of action. What are the psychological processes that drive you to do something? Then you can do it either within an action outcome, control over behavior process, or stimulus response, a skill. It doesn't matter. What matters is what drives the behavior to start with. Yeah?
So could drug taking be a skill though? Like say if you're injecting drugs, like there's a lot of steps involved and you have to have skill like finding a vein and stuff like that.
Yes.
Isn't that a skill?
That's a skill.
Yeah. That's
So
also
the effortlessness and the speed, these are features of skills. So that's why I told you skills and habits are built together. But skills are about performance. You don't care whether you can inject your drug well or not when you're seeking it. When you engage into seeking the drug, you're seeking the drug. And what actually made you engaging into seeking the drug? That's what matters. Either you wanted it or something in the environment drove the behavior. Then you can have skills, no skills, it matters less. But this is, I think this is the key question about nicotine addiction. If you ask me to smoke, I've never smoked a cigarette. I smoke cigars. But even when I was 3, if you asked me to smoke, I knew how to do that. Before I was exposed to the drug, I knew how to take the drug. I didn't really know, and I still don't really know how to actually self-inject heroin. It might be one of the reasons, one of the psychological mechanisms whereby nicotine is that addictive, because you've already implemented the habits before you've been exposed to the drug. You know exactly what to do, where to find it. You know the effect because of empathy processes. You know people when they smoke. Everything is there. When the drug kicks in, perhaps everything was ready for the drug to push the habit. It's not necessarily the case with illegal drugs, not even alcohol, because the way we drink alcohol is the way we drink water. Smoking is a specific behavior that is associated with nothing else but taking drugs. So that might be one, and you know, and we have no issue saying that children that were born 10 years ago, they were born with, you know, iPad, and they know that perfectly. It's part of their world. Cigarette is the same. So it might make a difference psychologically. So let's go back to habits. These behaviors are inflexible. They are not sensitive to manipulations of the representation of the motivational value of the outcome. And they are neither sensitive to manipulations of the representation of the causal instrumental relationship. Is that clear for everyone? No? If my behavior is habitual, because it's been initiated by stimuli in the environment and it's not self-initiated, I have no representation whatsoever before I do my action of its consequences. Okay? Which means that now, if I want to know whether a behavior is habitual or not, I've got to play with the subject's representation of the consequences of its behavior. Right? How would I do that in rats? I would have rats pressing levers. At what point, I would play with the motivational value of the reinforcer and ask the animal to tell me what they think they're working for under extinction. Under extinction, the reinforcer is not there. So what supports extinction is the representation the animal has of what he's working for. Either of the relationship between this is what I'm doing to get what I'm getting, or this is what I'm doing to get how well I'm going to like what I'm getting. Yes? So the first here, I'm going either to pre-feed the animal before the session. I've had my full, and now I'm not going to work for something, I'm just having my full. So goal-directed behavior, if you do that, the animal won't press under extinction. If the behavior is habit-based, they would still press under extinction. Now, how to do that? I'm going to proceed to a contingency degradation. So the animal expresses, under, you don't know, variable interval 30 seconds or FR5. And eventually, during these sessions, food is coming without any instrumental response. So there's no way I'm going to work because food is coming from the sky. So goal-directed actions, animals stop responding. Habit-based behavior, animals continue responding under extinction. Okay? And the same in humans. What's very important is that habits are more sensitive to Pavlovian influences than action outcome processes. This is a key element. Pavlovian conditioning and instrumental conditioning are differential learning processes, but they are heavily interacting. Pavlovian conditioning always influences instrumental conditioning, always. And habits are much more sensitive to Pavlovian influences than goal-directed actions. Have you heard about Pavlovian to instrumental transfer? A bit?
I would like to hear more.
So it's been popularized by Yin in the field of addiction. Yin suggesting that it was the mechanisms, one of the mechanisms whereby people would relapse after exposure to cues. I would slightly disagree with Yin, but he's partly right. What PIT, Pavlovian to instrumental transfer, is: first, a rat is placed in a box, and he receives pure Pavlovian conditioning sessions. A light shows up. 5 seconds later, a pellet comes in the magazine. The animal has nothing to do. And he learns that the light predicts the pellet. Okay? Then in the second phase of the conditioning session, the animal is subjected to instrumental conditioning. He learns that pressing a lever gives access to a pellet. But there's never, ever, ever any presentation of the light. Okay? Eventually, after 2 weeks of training under the instrumental conditioning setting, the animal is trained under extinction, so he stops responding, and then is presented non-contingently with the light. And as soon as the light is illuminated, the rat starts responding. Switch off the light, the rat stops responding, and so on. That's beautiful. The point is, it only works if the initial level responding is 0. PIT doesn't exist if the animals behave. Okay? This is a technical limitation. On the other hand, there's another psychological process that is dependent upon Pavlovian instrumental interactions that is called condition reinforcement. And we're going to discuss a lot about it, condition reinforcement. Condition reinforcement is the use of a conditioned stimulus as a reinforcer. I've been exposed to a Pavlovian context in which the light predicts the pellet. Okay? And then I'm exposed to any instrumental action, conditioning, and I'm pressing, and eventually, instead of the pellet, I'm given the light. And I would go on pressing for the light. Okay? Or I would learn a new instrumental action that is reinforced only by the light. If, by definition, I learn an instrumental action reinforced by the light is a reinforcer. This is acquisition of a new response. This is purely condition reinforcement. And condition reinforcement is the mechanism through which drug addicts can forage for their drugs for weeks or for days, because there are features in the environment that tell them you're getting closer to your drug. Okay? And what is very important, and this is, I think, a major element, and I would agree with George Koob about that, habits are triggered by stress. So every single drug that increases the activation of the stress system, thereby opening the window to the opponent process and allostasis, whatever you want, would facilitate the development of habit. Yep.
All drug habits or all habits are triggered by stress?
All habits are triggered by stress. Drugs only hijack the system, as they do with Pavlovian conditioning, as they do with everything. Okay? And neurobiologically, habits are dependent upon the dorsolateral striatum. And this is the first neurobiological data I'd like to show you. This isn't from my work, but a very nice work showing that control or stress animals are trained under ratio schedules of reinforcement, and they learn perfectly in their behavioral performance. It's exactly the same. Though one group is stressed, chronic stress, and one group is not stressed. The authors here have tested under extension the sensitivity to the evaluation of the outcome, either early or later on in training. And what they've shown is that in the stressed animal, the evaluation of the outcome, okay, resulted in a complete abolishment of instrumental responding under extension. That wasn't necessarily the case in stressed animals. And this actually happened in both early and late stages. And what's interesting is that these stressed animals that resist to the evaluation of the outcome, so they're habit-based, they have a decrease, they display a decrease in dendrites in the dorsomedial striatum and an increase of these dendrites in the dorsolateral striatum. The dorsomedial striatum together with the ventral striatum support goal-directed responses, and the dorsolateral striatum supports habit-based behavior. So you have, let's say, complete retraction of your ears in one structure and a complete spread of your ears in the other one. I'm listening carefully about what's happening when I'm habit-based in the dorsal striatum. So there is a functional balance within the dorsal striatum between the dorsomedial and the dorsolateral. And habits depend heavily upon the dorsolateral. So now, this is the gold standard model of addiction. One rat here in a self-administration box. He has to press 5 times on the lever. And actually, he's already taken cocaine, so he does a little bit of stereotypies with his head. And when rats take cocaine, it's not like food reward. They not stick to the lever. They have to move in the box because of psychomotor activity effects of the drug. But eventually, the animal will reach that stage at which he would press 5 times on the lever and get his drug. I think we are. Here we are. He received the drug-assisted stimulus, and now cocaine, 0.8 mg per kg, is delivered to its bloodstream. That's a very good model of drug taking. But that's it. And the main question is, why is this rat pressing this lever? Is the animal pressing the lever because he wants the drug, or is he pressing the lever because every single time he's put in his box, he's got nothing else to do than pressing a lever, and actually he's got drug in there? That's the key question. But definitely what matters is this is nothing of relevance with regard to addiction. This is an animal model of drug taking. I like whiskey. I drink whiskey pretty much every day. And from the very first day I've had my first glass of whiskey, I've had LTP in my ventral striatum. I've been exposed to drugs. Nevertheless, every day when I have my glass of whiskey, I have that, never more than that. The day I start sipping again, I'll be worried. But so far, I control perfectly. I'm a good model of alcohol drinking. I'm not sure I'm an alcoholic. Perhaps this is denial. Perhaps, you'll tell me, Guy, I'm sorry, but definitely not. So let's going back to the psychological processes that support the initiation of the action. A rat would learn to lever press to get cocaine. This is instrumental learning. Every single time he receives the drug, there are stimuli in the environment that predict and are associated with the delivery of the drug. The animal would actually develop Pavlovian conditioning, and there are Pavlovian instrumental interactions in the control over behavior. So there are two possibilities. First, I want my drug. I'm a rat, and I can have a representation of the outcome. I want my drug, and this drives my behavior, and I'm going to press the lever. Or I'm in this room, and there's this slide that has been associated with the drug. This tells me, go and seek the drug. Even if I don't have the drug, I would do that. Okay? So drugs facilitate habits. So after 2 weeks of instrumental responding for alcohol, rats would show resistance to the evaluation. After 8 weeks, they would show insensitivity to the evaluation, whereas the same training for sucrose, they would still show sensitivity to the evaluation for sucrose. So alcohol by itself facilitates the emergence of habits. And if you look at the time course, after 1, 2, 4 weeks, there's an increase in the magnitude of insensitivity to the evaluation that shows you that the animal initially has a representation of alcohol before it presses, and then it's the context that drives completely its behavior. This guy who's been asked to blow into this machine, and he tries drinking. That's it. The big issue we have is that the evaluation of outcomes is based upon ingestive reinforcers. You would pre-feed, you would render the animal sick. How would you devalue cocaine? Put an antagonist. The animal would press more to displace the antagonist. How can you devalue cocaine? And actually, sorry? The vaccine would have the same effect. Unfortunately, the vaccine is like pharmacology. So it would have the same effect. So the team of Schippenbergs has had this nice idea of using the seeking-taking chain schedule. So in this schedule, the animal first presses a lever for, let's say, a fixed interval, and then upon this fixed interval is passed, another lever comes into the box, and the animal presses this lever to get the cocaine. So first the animal presses for one lever that is associated with cocaine delivery. And what they did was they tested whether extension of the second lever had an impact onto the responses on the first seeking lever. If it has an impact, it means that the animal presses the first lever because he has a representation of access to the second one, and the second one gives cocaine. If it has no impact, then the behavior is habitual. And they show that after 15 days of training, rats are highly sensitive to the evaluation, to the extension of the lever, and after 40 days, they are absolutely habitual. They don't care whether you give them the drug or not. They press. What's interesting is that we've been running, I think I've been running 10 studies like this right now. And the emergence of compulsive behavior, I'll come back to that in the second part of my lecture, but the emergence of compulsive behavior takes at least 45 to 50 days. And habits appear before that. So it led us to believe that habits is a necessary step towards development of compulsivity. So briefly, what is this? I take a cohort of animals, give them self-administration, FR5, but every single day, the basic session is 40 minutes access to the drug with a specific stimulus that says the drug is available, 15 minutes where the drug is not available, and there's another stimulus that says the drug is not available, 40 minutes access to the drug, 50 minutes no drug, 40 minutes access to the drug. Initially, all the animals learn to press when there's a light that says the drug is here, and not to press when there's the light that says there's no drug. And eventually, after 30 days, some animals start pressing in these no drug periods. They just can't stop seeking the drug. And these animals eventually would be those that show the highest scores in the progressive ratio schedule reinforcement that is supposed to actually to measure motivation, but we can discuss about that. It's not clear. And there would be those animals that resist to punishment.
So David, why do you say that's not goal-directed? Because everybody's testing the environment all the time for changes. Because they've learned that sometimes things aren't what they expect.
So that's why actually that's why you use habits. You do what you got to do so that you can monitor the environment. And if there are changes, then you update what you're doing.
Yeah, right.
Yeah.
But there's two ways. I mean, you can either enhance the habits or decrease the ability to.
Yeah, Hugh Garavan has very nice studies in humans showing this balance and how you can regain control of a habit depending on how the imbalance between habit and goal-directedness is important. So you're right, you can have either a decrease of one or an increase of the other. But what matters is the contrast between the two. So we select the animals that display the three behaviors. And you have 3 criteria rats that we believe are addicted and 0 criteria rats that are not addicted. But I'll come back to that later. The point is, when you put together, it's been shown that there is a transition from impulsive behaviors to compulsive behaviors in the development of drug addiction. Initially, I would take drugs in a framework of risk-taking. Everybody knows it's a risk to take a drug. You know, but eventually, in a certain context, I would overcome my inhibition and I would take my drug. By definition, this is impulsivity. Risk-taking is a facet of impulsivity. But eventually, I would end up in a compulsive behavior. And we suggest that it comes in parallel with a transition from goal-directed behavior that undergoes incentive sensitization. That's important. how much Pavlovian influences would control your behavior. Opponent processes, these adaptations to chronic drug exposure would still drive your behavior, because when I feel bad, the effect of my drug not only would give me euphorism, but would relieve from the fact that I'm feeling bad. So that's important. And on the instrumental side, habits would develop, such that at the end, incentive sensitization plus opponent processes would interact with the habit system to render this behavior like an incentive habit-based. And these incentive habits, once the prefrontal executive control over behavior is lost, are, I think, the ground on which compulsivity is built or developed. Okay? So neurobiologic, yep.
So do you think people who are more vulnerable to drug addiction just transition from this habit to the habit incentive much more quickly?
You're hard. So it was supposed to come later in the course, but no. I'll answer the question right now. Where are we? This is a key question. Okay, so let's start here. In 2008, I tested whether impulsivity predicted the transition to compulsivity. That was the framework on which Kub and Lemoine developed their hypothesis. And I was lucky that I did my PhD in Michel Lemoine's lab, and I ended up in Cambridge, where they were working on impulsivity. So, here we are. And we demonstrate that impulsive responses in the file choice side reaction time task, but if you don't mind, we'll go into the details of this task later on. Basically, you ask the animal to wait for stimuli in the environment to tell them what is the good behavior to perform. And sometimes, if you ask some of them to wait a bit longer, they're going to try before they know what to do. That's a premature response in the task, and that is impulsivity. A facet of impulsivity, it's motor impulsivity. And this has been shown to be dissociable from attention, perceptive responses. This is pure impulsivity. And this impulsivity has been shown by Jeff Daly to be associated with a decreased D2, D3 binding in the ventral striatum prior to any drug exposure. And these high impulsive animals would escalate cocaine self-administration more than low impulsive animals. And we've shown here that high impulsivity predicts the transition to compulsivity in the 3-quaternion model. And that high impulsivity here clusters with addiction and is orthogonal to locomotor reactivity to novelty, the Piazza HR-LR model that has been shown to predict increased propensity to self-administer drugs. HR rats would self-administer drugs at doses at which LR rats feel nothing, but they've got nothing to do with addiction. Okay, so first, the propensity to use drugs is not necessarily a good marker of the vulnerability to switch from controlled drug use to compulsive drug use. Okay, so then, would high impulsive animals facilitate, actually in impulsive animals, the transition to habit-based behavior, so the recruitment of the dorsal striatum, is it facilitated? That's your question, isn't it? So first we performed an in situ hybridization experiment in these animals. Rats were exposed to either drug naive, either 10 days of cocaine self-administration or 50 days of cocaine self-administration. In these conditions, we know promote compulsivity. We didn't test compulsivity because we knew we would have differential responses, and then this would have completely disrupted the potential results we would have had. Okay? Nevertheless, high impulsive animals here on the left, after 10 days of cocaine exposure, they tend to show lower decrease in D2 receptors in the ventral striatum and the VTA as compared to low impulsive animals. So the size of the cycles are the magnitude of the effect. So low impulsive animals show facilitated transition here. Oh, and I don't have the long axis. I'll show you the long axis later. So this is, sorry, drug naive after a week, after 10 days of exposure, low impulsive animals show a decrease in the ventral striatum. High impulsive animals do not show that. And after chronic exposure, I'll show you on another slide, here there is a decrease in both sides in the dorsal striatum, in the core of the nucleus accumbens, and in high impulsive animals, you only start seeing the decrease in the shell and in the DLS. So there is a shift in the transition. Okay? And now if you causally probe whether this is the case or not, and you check the recruitment of the dopamine transmission into the dorsal striatum in the support of drug-seeking behavior. So high impulsive animals are here, low impulsive animals are here. If you inactivate dose dependently with alpha-frequentixol, 5, 10, 15 microgram per side of alpha-frequentixol, 5 minutes prior to drug seeking, when the behavior is goal-directed, you have no effect of these manipulations in either high or low. We knew that already. When the behavior is intermediate, so the animals have been trained under a ratio schedule of reinforcement that we know starts triggering habits, but not quite completely. Actually, high impulsive animals are insensitive to the manipulations of dopamine transmission into the dorsal striatum, but low impulsive animals start showing a sensitivity that is pretty much of the same magnitude of the sensitivity every single rat shows when the behavior is habitual. So your question is impulsivity facilitating the development of habits? The answer is no. It's the other way around. That's actually, so this is under re-revision in biological psychiatry, and we thought we would have had a hard time because that's what people would expect. But the point is, why would people expect that? Because, you know, vulnerability comes from failure in the cortex, intrastriatal mechanisms, and what we've shown here is that actually vulnerability interacts with the transition to the dorsal striatum in a way that is not in the striatum. And the point is, I told you initially that once incentive habits have developed, you would develop compulsivity if you lose control over that. So how impulsivity interacts with habits in promoting addiction, to promote addiction, is perhaps through a loss of control over the habits, not in facilitating the development of habits. Another key aspect, that because you need much more exposure to drug and to condition enforcement in high impulsive animals to recruit the dorsal striatum, it means that your synaptic plasticity processes are perhaps less flexible in these animals. And you need to overcome a kind of resistance to plasticity, drug-induced plasticity. And perhaps, and this is what we're starting to investigate now, once these animals, high impulsive animals, have reached that stage, how fast you reach the stage doesn't matter much. Whether you reach that stage and you can come back is what matters. And we are now investigating whether once these have reached that stage, these ones can easily come back here, or whether these ones can never come back here. This might be the case. Is that how fast you go is once you're there, can you come back? Yeah? So I've got to find where I was now. I was here. So the conclusion of my talk, I'm giving that to you now, because then it's going to be tough, I think, is that drug-seeking behavior initially is dependent upon a network that involves the orbitofrontal cortex, infralimbic and prelimbic cortices, the dorsomedial striatum, and the ventral striatum. This is goal-directed behavior. This is managing alternatives in the environment and motivation. This is dealing with choices. And this actually corticostriatal network is controlled by the basolateral nucleus of the amygdala, and its reciprocal influences with dopaminergic processes in the VTA. That's the network that subserves recreational drug use, regular self-administration. When the behavior is habitual, what we've shown is you've got a complete disengagement of the dorsomedial striatum, a major recruitment of the central nucleus of the amygdala, and actually two amygdalostriatal pathways driving the behavior. A BLA core spiraling loops. Have you heard about Susan Haber's spiraling loops in the striatum? We'll come back to that. Spiraling loops, and another pathway, central nigra DRS. And you need these two pathways to drive cocaine-seeking habits. How did we demonstrate that? So that's the story I'm going to show you now. So first, we were very interested in the fact that drug exposure triggers adaptations in the brain that are initially restricted, if you consider the striatum, to the ventral parts of the striatum and the medial parts of the striatum. So this is that binding in monkeys, self-administering cocaine. I think this is after 3 months, and this is after 3 years, perhaps. So you've got an increase in that binding in the ventral areas and medial areas of the striatum, and after 3 years, you've got a spread, complete spread of these adaptations to the dorsal areas of the striatum. Okay? And this can be put in parallel with the increased dopamine transmission in the dorsal striatum in drug addicts that crave cocaine. The same spread from the ventral to the dorsal striatum in terms of neurobiological adaptations to cocaine has been shown with regards to the D2 receptor density in monkeys, where you have decreased D2 receptor density initially restricted to the ventral areas, and that progressively recruit the dorsal parts of the striatum in monkeys. And we've shown that this was the same in rats with in situ hybridization. Initially, there's a decreased D2 receptor levels, mRNA levels, in the shell of the nucleus accumbens, and after 50 days of self-administration, it spreads to the nucleus accumbens and all the dorsal territories of the striatum. So there are indeed adaptations that take place initially in the ventral striatum and spread to the dorsal striatum. How can you account for this spread from ventral to dorsal? One potential neuroanatomical substrate was the new, not brand new now, but actually the new functional anatomy of the basal ganglia that was described by Susan Haber. Susan showed that GABAergic neurons in the shell that receives projections from the VTA send back projections to these neurons in the VTA in a feed-forward loop. But they also send projections in neurons in the VTA that are more lateral, and these more lateral neurons in the VTA project to more lateral neurons in the striatum, and so on, such that dopamine transmission in the shell can eventually control what's happening in the dorsal striatum. Okay? So if this neuroanatomical substrate is indeed the one through which a recruitment of the dorsal striatum occurs, then we might be able to test it by manipulated intrastriatal processes. But for that, we need a task that measures drug seeking without any drug infusion. We want to check craving is not about taking the drug. It's about seeking the drug. Drug addicts spend 90% of their time seeking the drug and only 3% of their time taking the drug. So we need to dissociate seeking and taking responses. And how to measure seeking and taking? You need to have seeking sequences for long periods of time. And we want seeking responses under the control of stimuli in the environment, so under conditioned reinforcement. And this is what you can have with second-order schedules of reinforcement. This rat is going to press the lever for 15 minutes. Every 10 lever presses, it receives an illumination of the light. And look at the behavior. The rat is highly interested in the light. Okay? This rat is going to press 300 times this lever in 15 minutes for nothing. Nothing at all. Because fixed interval schedules of reinforcement are built such that the amount of responses during the interval doesn't predict access to the reinforcer. Because access to the reinforcer occurs upon the first lever press on the lever once the 15 minutes have elapsed. So if the rat was clever, we would just wait 15 minutes, presses once. In terms of behavioral economy, that's what you would expect. Presses once, he gets his reward. But rats, monkeys, dogs, humans, they start anticipating. They can't wait that long. And the point is, there's a complete dissociation between responses and outcome. Okay? But on top of that, every 10 lever presses during these 15 minutes, the animal receives the conditioned reinforcer. It receives the light. And in that case, this is what happens. Before we introduce the light as a conditioned reinforcer, so the animal presses 15 minutes. After these 15 minutes, he receives cocaine plus the light. In average, rats would press 70 times for nothing. Okay, they have to press once after the 15 minutes have elapsed to get the drug, but they would press 70 times. Now, every 10 lever press, you introduce a contingent presentation of the light. The animal would press up to 150 times. They are self-administering light. Yeah, you agree with that? The difference between the yellow bar here and the pink bar here, this is the effect of conditioned reinforcement. How much contingent presentation of Pavlovian stimuli support high levels of instrumental responding. And if you train these animals under this schedule for 2 weeks, then you take the cocaine out, and the animal will go on pressing forever. You take the CS out, the animal stops pressing completely. Its behavior is completely reinforced by the stimulus from the environment. Okay? So these were results to show the effect of CS submission, but I ask you to believe me that that's the case. So now the psychology of this and the psychobiology of this process. For the acquisition of drug-seeking responses, you need an intact basolateral amygdala. Obviously, because there's a Pavlovian component in there, and CS-US associations that are necessary for Pavlovian conditioning are built and managed in the basolateral nucleus of the amygdala. So you need the Pavlovian component and the basolateral nucleus of the amygdala to acquire cue-controlled drug-seeking behavior. Okay? And you also need an intact core of the nucleus accumbens to acquire cue-controlled drug-seeking behavior, not the shell. The shell is necessary for the acquisition of self-administration, fair enough, but not for the development of cue-controlled drug-seeking behavior. And we know that the BLA and the core interact massively. And Patricia Di Ciano in Barrett's lab a few years ago demonstrated that if you infuse dopamine into the core of the nucleus accumbens or the BLA, there is an effect of dopamine blockade in the BLA on responses in early stages of cue-controlled drug-seeking behavior. However, if you block glutamate transmission in the core, but not the BLA, you have a decrease of early established cue-controlled drug-seeking behavior. So it seems that dopamine in the BLA and glutamate in the core are necessary to support the acquisition of this behavior. Now, how do you probe whether they indeed functionally interact in promoting this behavior? You would perform what we called functional disconnections. So this is an example for intrastriatal processes, but the principle of functional disconnection is I have two structures that have been shown independently to be involved in a specific behavior. Okay? So when I take out one structure on both sides of the brain, I lose my behavior. When I take out the other structure on both sides of the brain, I also lose my behavior. Now there are two potential explanations. First, these are additive mechanisms, and the two structures don't interact. In that case, if I take out my structure on one side of the brain, this one, and this one on the other side of the brain, then the intact structures should support at least a part of the behavior. Now, if my two structures are serially linked, the product of structure here would be the substrate of this one in the emergence of the behavior. So if I take one structure on one side of the brain and the other one on the other side, I take out the entrance and the end of the loop on both sides, and I should lose my behavior. Yeah? That's exactly what was shown. You infuse vehicle in the BLA, vehicle in the accumbens. This is the level of responding. Then either dopamine antagonist in one and glutamate antagonist in the other, you don't lose your behavior, and you functionally disconnect the two structures. Glutamate antagonist in the core, dopamine antagonist in the BLA, then you completely lose your behavior. So you need functional interactions between the BLA and the core of the nucleus accumbens in the development of cue-controlled drug-seeking behavior. However, when this behavior is well established, as I told you, after 2 weeks, now in microdialysis, Rutukoito demonstrated that rats are seeking cocaine for 15 minutes. Actually, here it was 20 minutes, but it doesn't matter. And within these 20 minutes, you present non-contingently the drug-related stimulus. Okay? The animal doesn't seek it, you just present it. No effect on the pain transmission in the striatal. But then you start the second-order schedule of reinforcement procedure, in which every 10 lever press results in the presentation of the light. And this is the blue sequence here. And there's a massive increase in dopamine release into the dorsolateral. Then when you stop the behavior and you present once again non-contingently the conditioned stimulus, there's no increase in dopamine. So this shows that when the animal is pressing for the conditioned reinforcer, there is an increase in dopamine transmission into the dorsal striatum that has nothing to do with Pavlovian conditioning on its own. Otherwise, we would have dopaminergic responses to non-contingent presentation of the conditioned stimuli. Is that clear for everyone? Yeah? So this was the first correlate of recruitment of dorsal striatal territories in drug-seeking behaviors. After that, Luc van der Schoen in Barry Everitt's lab demonstrate causally that if you blocked dopamine transmission into the dorsolateral striatum by infusing bilaterally alpha-flupenthixol, a mixed D1, D2 antagonist, into the dorsal striatum, you would dose-dependently decrease the behavior only provided it is well established, always after at least 2 weeks of training. So that was the causal demonstration that indeed, when the behavior, cue-controlled drug-seeking behavior is well established and habitual, there's a dopamine component into the dorsal striatum. And this resonates well with Schippenberg's story I talked about. When the animals resist to, remember, they resist to extension of the taking lever. Now you inject lidocaine into the dorsal striatum and you render the behavior that was resistant sensitive once again to the extension of the taking lever. By inactivating the dorsal striatum, you re-express goal-directedness. So when the behavior is habitual, it's not as if goal-directed behavior has been washed out. It's there, but habit-based behavior just takes over. Okay, now you take out habit-based behavior and goal-directed behavior comes in. That's the theory, because that's not exactly what we have, and I have an explanation for that. So these are the principles of the demonstration that Susan Haber's loop might be the mechanism through which you recruit the dorsal striatum. We've been investigating whether blockade of dopamine transmission on both sides of the dorsolateral striatum had the same behavioral effect as combining a unilateral lesion of the core of the insuccubans and a contralateral blockade of dopamine transmission into the dorsolateral striatum. Okay, the principle of disconnection, but within the striatum. We made sure that lesioned animals, okay, acquired conditioned reinforcement to the same extent, the same way as control animals. So the unilateral core lesion on its own had no effect on the acquisition of cue-controlled drug-seeking behavior. After 3 weeks of training, we took the control animals that had no lesion, and we performed a unilateral infusion of alpha-flupenthixol into the dorsolateral striatum. And we show that this manipulation has no effect on well-established cue-controlled drug-seeking behavior. So 2 manipulations that are ineffective on their own, you combine them, thereby functionally disconnecting the ventral to dopamine transmission into the dorsal striatum, and you end up with a dose-dependent decrease in cue-controlled drug-seeking behavior that is of the same magnitude as bilateral manipulations of dopamine to the dorsal striatum. So this experiment, so we made sure that this didn't occur early on in training. In these same animals, we had them pull a chain to get a sucrose solution, and actually they were not sensitive to DLS manipulations. So this experiment, so that's the effect early on in training, no effect of this manipulation. This experiment suggests that indeed there are ventral to dorsal functional interactions that drive the behavior to be controlled by the dorsolateral striatum. But the control of behavior by the dorsal striatum depends upon the integrity of the ventral striatum. This fits well with the actor-critic model of the basal ganglia. The nux accumbens tells the DLS what to do. The point is, and I'll show you later on, there is a nice molecular psychiatry paper that came in December last year in former heroin addicts. There is an abnormal functional synchronicity between ventral and dorsal striatum in drug addicts, so that the actor-critic model doesn't work. It's just an actor. The ventral doesn't teach the dorsal anymore. It is just a pathway through which impulses from the BLA reach the DLS. It doesn't teach anymore. So there's no critic to be performed. I showed you some results in food rewards and stress and habits, and I showed you that spines in DLS increase, whereas they decrease in the DMS in habit-based behavior. That's Jen Murray results. So Jen tested whether we also observed a dorsomedial to dorsolateral shift in the control over behavior in cue-controlled drug seeking. So she tested the animals early on in training when the behavior is goal-directed, and she shows that neither in the DMS nor in the DLS you would find any effect of dopamine manipulation. Sorry, in the DLS there's no effect of early performance of seeking behavior, and there's no effect on drug self-administration per se. Whereas early on in the posterior dorsomedial striatum, there is a dose-dependent decrease in cocaine-seeking responses that has nothing to do with cocaine taking. Cocaine-seeking responses respond to some treatments that are absolutely ineffective on cocaine-taking responses. Drug self-administration, that is, let's say, a consumatory response, is dissociable from drug-seeking responses that are anticipatory, preparatory responses. However, when the behavior is well established, it's exactly the opposite. You would have a massive effect of a manipulation of the dorsolateral striatum that is not reflected in taking, and you have no effect of manipulation in the dorsomedial striatum. So there is indeed a shift when the behavior becomes well established from the dorsomedial to the dorsolateral striatum in the control of drug-seeking responses. So we've been talking about intrastriatal shifts. There is a ventral to dorsal shift at the same time as a dorsomedial to a dorsolateral shift. So it goes from a ventral dorsomedial to a ventral dorsolateral shift. Okay? But what drives that? What could be driving these shifts? So we've been interested in the control by the amygdala. I initially mentioned that the amygdala was necessary for the acquisition of cue-controlled drug-seeking responses. And Packard had demonstrated that if you inject stress-related peptides in the basolateral nucleus of the amygdala, you enhance habit-based responding in spatial navigation, for example. So Jen, once again, she tried to disconnect the basolateral and central nucleus of the amygdala from the dorsolateral striatum. Okay, so she performed a unilateral lesion of either the BLA or the central, and she combined this unilateral lesion to dopamine receptor blockade in the dorsolateral striatum in the contralateral side of the brain. And she shows that in doing so, you end up with a dose-dependent decrease in cue-controlled drug-seeking behavior in the basolateral nucleus of the amygdala and the central. So the two nuclei in the amygdala are necessary to recruit dorsolateral striatum-dependent habitual drug-seeking behavior. That's nice, but the wrong side of the story, or the bad side of the story, is that the BLA projects to the central. So very likely, this effect can be entirely attributed to, actually this effect could be attributed to the lesion, actually to a lack of BLA effect. But remind that we perform the lesion prior to training. We want to prevent the recruitment of the loop on this side of the brain. So this is the work performed by my beautiful wife, who is working in my lab. She's an electrophysiologist, and she's checked whether in drug-naive animals, the BLA and the central control differentially the activation of medium spiny neurons in the DLS. So for this, she designed a setup in which she stimulates the motor cortex 1, that is the major glutamatergic inputs onto the medium spiny neurons in the DLS, which are under anesthesia very silent. She records these neurons, and she approaches her extracellular electrode to the neuron such that she ends up with a nice artifact, a nice action potential post artifact. Then she sets the probability of response of this neuron to 50%. Why does she do that? Because if you want to see an increase or decrease, if you're 100% or 0%, you can see only on one way, not the other. And then, once she's set up this, she starts investigating the effect of a stimulation of the basolateral of the amygdala on the responses of medium spiny neurons to a stimulation of motor cortex 1. Okay? So what is shown is that first, if you stimulate the amygdala alone, the neurons in the DLS don't respond to BLA stimulation alone. Obviously, there are no direct relationships, no direct projections between the two structures. So it would have been very unlikely that we found that. But then, what is shown is that if you stimulate the BLA with various latencies before stimulation of M1, you end up with various responses, various populations of neurons. Some neurons here are upregulated by the pre-stimulation of the BLA. Some neurons are non-regulated, and some neurons are down-regulated. So indeed, this cortex here, that is an allocortex, very remote from the cognitive areas and the cognitive networks to which belong the DLS, the basolateral amygdala controls the response of medium spiny neurons in the DLS to stimulation of the motor cortex. I think this is the first demonstration that limbic areas, such as the amygdala, control cognitive and motor areas, such as the cortex. What's interesting is now, I told you she's been investigating different latencies between BLA and M1. So motor cortex stimulation alone, 50% responses. BLA stimulation alone, no responses. And for the short latencies, no effects. Why is it important to probe short latencies, 1 or 5 milliseconds? Because these short latencies test whether the system is monosynaptic or not. If we had effects with short latencies, this would mean that we have direct BLA dorsolateral striatum projections. And it would be a pain because nobody has ever demonstrated that. There are direct BLA posterior dorsomedial striatum projections, massive ones. That was your question. Not a single one with the dorsolateral striatum. If there were any, they would be in the patches. If there were. There are definitely none in the dorsolateral striatum. A lot in the posterior dorsomedial one. Now, if you look at longer latencies, between 100 and 300 milliseconds, this is where you end up identifying the up, down, and non-regulated neurons. So you need several synapses for the effect of the BLA to reach the DLS. And that fits pretty well with the notion of the loop. Okay? The fact that there are different latencies here might suggest that depending on the animals, either the loop is longer, shorter, more synapses or not, we still don't know. But the fact is, there's a control that might go through the loop. And it might not be going through the central amygdala, because we know that the pathway from the BLA to the DLS through the central amygdala requires BLA central, central nigra, nigra DLS. That's a very short latency. And we found no effect of short latencies. But nevertheless, this didn't demonstrate whether the control of BLA over DLS responses, is it already one?
Yeah.
I can't believe that.
I know, you've been going.
What time should I stop?
About half an hour ago. That's all right, we can have a break.
Holy crap, I'm sorry.
It's all right, you were really into it, it's good.
Yeah, I apologize. I was just, I saw the time there. So the point is, in order to make sure the effect of BLA control over DLS go through the core, Aude combined these manipulations with an inactivation of the core. So she infused into the core a mix of AP5 and I don't remember what. She blocked glutamate transmission in the core. And in the same neurons in which she showed up or down regulation, she recorded once again, according to a Latin square, whether an inactivation of the glutamate transmission in the core had an effect on the responses. And that's what she got. Upregulated neuron, downregulated neuron. After the same neuron, after inactivation of the core, you lose the effect of BLA stimulation on the response in the dorsal striatum. Completely. And you have obviously no effect in non-regulating neurons. So overall, what does it tell us? This tells us that indeed, initially the behavior is goal-directed. It depends upon the posterior dorsomedial striatum, the ventral striatum, and the basolateral nucleus of the amygdala. Pavlovian components, goal-directed behavior, condition reinforcement emerges from their interactions. And when cue-controlled drug-seeking behavior becomes habitual, then there is a dorsomedial to a dorsolateral shift. This dorsolateral striatum is under the control of the ventral striatum, but the ventral striatum only then drives the information from the BLA to the DLS through these loops. But this informational loop here is not necessarily sufficient to trigger the control over dorsal striatum, because you need, on top of it, a central NIGRA DLS loop to trigger the behavior. And this is where incentive habits happen. This is opponent processes, stress-related and arousal-related processes, driving massively information through the NIGRA to the DLS, on top of a drug-related component that is Pavlovian here that drives through the ventral striatum, the dorsal striatum. So you have an informational route from BLA to core loops DLS, and actually enhancing route from the central to the NIGRA and the DLS. Incentive sensitization is here and here. So everything can be put together. All the adaptations and the affective responses to the drug in the emotional cortices here can control the recruitment of dorsolateral striatum-dependent processes.
So that's exactly my question. We've been talking a lot about ventral structures. So how does this dorsomedial, dorsolateral difference map on to the differential cortical inputs that they're receiving?
So the posterior dorsomedial striatum receives major inputs from the amygdala, orbitofrontal cortex. The patch here, there, receive projections from the infralimbic cortex. Here, there's only the motor cortex that drives. Somatosensory cortex over here, but here it's more the motor, and the medial dorsal nucleus of the thalamus. So there's a complete dissociation. The key question here is, fair enough, I think we start disentangling and understanding how subcortical processes can drive the behavior. But Linda Porino has also demonstrated that this medioventral to lateral shift that occurs in the striatum also occurs in the cortex, with alterations initially restricted to the orbital cortex and spreading up to the motor cortex. So the question is, are these transitions in the cortex driving or contribute to drive these ones? Are they additive processes? Are these transitions in the striatum driving the cortical ones? This we don't know. But definitely what happens when drug seeking is habitual is you can end up with either internal stimuli, an internal urge, or stimuli in the environment that drive a non-explicit, rigid automatic behavior. So you can relapse, not even thinking about your drug. Yeah? I stop here now.
Okay.
And I apologize.